Organisms reproduce themselves, and thus each species is perpetuated. Some simple plants and animals reproduce only by asexually duplicating themselves generation after generation, allowing little or no variation. But most organisms reproduce in a sexual union of sperm and egg by a process called fertilization. These invertebrate eggs are surrounded by sperm. In a sexual union, each parent contributes a special cell that has been formed through a unique process, meiosis. These cannot be ordinary cells because such a union would produce a new organism with double the normal number of chromosomes for that species. Chromosomes are the packages of genetic information needed to regulate both the individual cells and the whole organism. Each chromosome has in it numerous genes which are the units of information. For each species of plant or animal, there is a specific number of chromosomes in each cell. Horses have 64 chromosomes. Cows have 38. Cats also have 38. Fruit flies have 8. Humans have 46. The chromosomes vary in shape and size, but there are 2 of each type. They are paired. The members of each pair contain similar but not identical information. The members of each pair are homologous. All but the reproductive cells of higher organisms have homologous chromosomes. Cells with homologous chromosomes are diploid. Reproductive cells, called gametes, are different. They contain only half the full number of chromosomes, 1 chromosome from each pair. These cells are haploid. Gametes are produced in the reproductive organs of males and females. When a male gamete or sperm joins a female gamete or egg, the process is called fertilization. A fertilized egg called a zygote is produced. This new cell is diploid because it has homologous chromosomes, 1 set from each parent. The zygote will multiply by cell division and eventually grow and develop into an adult. Chromosomes are able to make copies of themselves. Here, a chromosome is seen after it has formed a copy of itself. These 2 copies, called chromatids, are normally identical to each other. This is a homologous pair, 2 similar chromosomes, each made up of 2 identical chromatids. Gametes are produced in a special kind of cell division in which the normal number of chromosomes is reduced to half that number. This form of cell division is called meiosis. In the process, each gamete ends up with a copy of one of the chromosomes of each pair. Here is a simplified representation of the process of meiosis. Outside the cell's nucleus is a centriole. The centriole divides and the 2 centrioles move apart. The nuclear material coils up into visible chromosomes. Many fibers connect the 2 centrioles, forming the spindle. The homologous pairs of chromosomes line up in the center of the cell. The chromosomes separate as the cell divides to form 2 new cells. The chromosome from each homologous pair ends up in each cell. The chromosomes uncoil as the new cells prepare for a second division. As the second division takes place, new spindles form. The chromosomes again coil and line up on the centers of the 2 cells. the chromatids separate and enter into individual gametes. 4 gametes have been produced. Each gamete is haploid, having half the chromosome number of the original cell. Gametes are produced in both plants and animals from cells that are segregated in an early stage of embryonic development from the other cells of the organism. These cells are located in specialized organs. In most plants, gametes are formed in the flowers. Pollen, the male gamete, is produced in the anthers. The eggs are formed in the ovaries. Pollen grains grow tubules which, in fertilization, extend to the egg. Placed in sugar water or honey, pollen tubules can be seen to grow. The union of pollen nucleus and the egg results in a seed, the beginning of a new plant. In animals, including humans, gametogenesis, or gamete production, occurs in the gonads. Males form sperm, while females produce ova, or eggs. Meiosis is a continuous process, but for convenience, it is usually examined as a series of separate events. As the cell prepares for meiosis, the chromosomes gradually become more visible. This is because the long, thin strands of the chromosomes coil back on themselves again and again until they form a thick structure. As the chromosomes coil up, slender fibers begin to assemble around the outside of the nucleus, forming the spindle. The nuclear membrane disappears. Each chromosome is made up of 2 identical chromatids. As the chromatids of homologous chromosomes lie side by side, there can be an exchange of genetic material. This has great significance in heredity. It is known as crossing over. The homologous chromosomes align themselves on each side of an imaginary equatorial plane. The chromosomes move toward opposite poles. Because of crossing over, some paired chromatids probably no longer carry identical genetic information. When the chromosomes reach the opposite poles, new nuclear membranes begin to form around them, and the chromosomes uncoil. Quite soon, the 2 cells begin to divide again. The chromosomes coil and are aligned around a new equatorial plane that is at right angles to the first one. The chromatids separate and go to opposite poles. The result is 4 new cells, each of which is haploid. They were formed by meiosis from one diploid cell. The nuclei reform, and new nuclear membranes develop. Eggs and sperm are produced by slightly different processes. In many species, including humans, many more male than female cells undergo meiosis. Up to 300 million sperm are present at any one time. In men, meiosis of one diploid sex cell results in 4 haploid sperm. In women, however, only one of the new cells will mature into an egg. The other 3 never fully develop. Only about 400 eggs are produced in a lifetime. On the average, 1 per month during childbearing years. From an hereditary point of view, it is important to remember that although the new cells share many genes in common, each cell is unique. Each contains a different combination of genetic information. How does this occur? In higher organisms, all cells except gametes are diploid. They have two complete sets of genetic information. These are the chromosomes of a human male. Why do organisms need two sets? Having two sets allows for the possibility of great diversity. To recognize why this is so, it is necessary to understand that these complementary sets of information are contained in the homologous chromosomes. Each chromosome has in it genes that are the units of information. Each gene is found at a particular point or locus on the chromosome. On homologous chromosomes, genes that code for the same thing, such as eye color, are located at equivalent points. These genes code for the same thing, but are not necessarily identical. One may code for brown eyes, and although the equivalent gene also codes for eye color, it may code for blue. Already there is some variety of genetic information. At the start of meiosis, when the homologous chromosomes pair up, they overlap and may stick together at the overlap points. Then, as they are pulled apart, crossing over takes place. Random segments are switched, and an exchange of genetic material occurs. As meiosis proceeds, these new sets of genes are segregated. This results in a shuffling of genes, a redistribution of sets of genetic information among the four haploid cells, gametes. Which set of genetic information goes to which new cell is a matter of pure chance. When fertilization, the union of a male and female gamete, takes place, the genes contributed by each gamete are reassembled in new combinations. Based on this knowledge of genetics, we have used selective breeding to change and improve our crops in order to better serve our growing need for food. And the process is ongoing. In a changing world with an increasing population, new and different varieties of crops are continuously needed. Crossing over, shuffling, fertilization. Each provides an opportunity for the formation of new gene combinations. And when all three means of introducing variety into gene combinations are considered together, the possible numbers of new combinations are vast indeed. So vast are the possibilities that each fertilization brings about a unique organism, a true individual who is similar to, but never exactly like, any other member of its own species.